We are recording the program so we can share it later with a wider audience. Please keep your microphone muted and your video off unless you're presenting. We are going to invite everyone to join us on video during the Q&A, the final Q&A. So we hope you stick around for that. There will be 2 presentations with time for questions between each presenter. And you can send those questions via the chat feature if you'd like. It is helpful if you hold those questions until the presenter is finished speaking. We're thrilled to have you all here for this discussion. And with that, I'm going to introduce our team captain, Cindy Brandt-Levin. Cindy is the FDMAS Alliance Vice President and the leader of Team FDMAS in the UPenn Orphan Disease Center's $1 Million Bike Ride. Cindy's championed this initiative since the very beginning and devotes significant time and energy to the organization and our mission beyond the $1 Million Bike Ride. We're so grateful to have her leadership and determination. Cindy, thank you so much.
Great. Thank you, Tova. Thank you, Tova, for organizing this presentation. You've done a fabulous job. Everyone who's here can see the passion that you lead as our community engagement leader, and of course our fearless leader, Adrian, who has taken on the reins of executive director and has brought this organization to new heights. I am privileged to be here and excited to hear about 2 of our awardees back from 2018, I believe this is, or 2019. With regard to our past $1 Million Bike Rides. I wanted to first just note that I believe, I can't see everyone who's on this video, but I believe we do have members of our Medical and Scientific Advisory Council, Drs. Len Cabin, Lillian Plotkin, Ed Chow, and of course, Dr. Michael Collins. Without the help of the members of these of our councils, we could not be where we are today in terms of helping with the research grants for the bike rides, as well as all of the work that they do for our organization. We also have some of our board of directors here who I am privileged to serve with. Ann Corvell, who will be our new president in the next session. Joyce Ellis, Sarah Healy, Melissa Kenny, and Kieran Yargalata. They're all here today as representative of our great volunteer board. And also we have here Lily Lagnew from Association MASFD, it's opposite of ours in terms of the title in France. And she has devoted considerable energy to the $1 Million Bike Ride initiative, donated personally from the organization and herself, and all of the work that she has done. And thank you, Lily, as well. I just wanted to note, as you'll see on this slide, this is our 8th year of partnership with UPenn. The last 2 years, we've only rode virtually, and I'm excited to go back and ride this year in person, and hope some of you will join us, whether virtually or for the ride. I believe we will hit the $1 million mark of fundraising for this great initiative this year. We're over $900,000 since I started the team back in 2015. I can't believe it. It's been that long. We've had 15 research grants awarded since its inception, and we're going to hear about 2 of them today. And without further ado, I just wanted to introduce Dr. Michael Collins, and I'm going to read his bio since I can sometimes mess this up. He is the senior investigator in the skeletal disorders and mineral homeostasis section of the National Institute of Dental and Craniofacial Research, and has devoted his career on the study of fibrous dysplasia and McCune-Albright syndrome, including close collaboration with our organization since founding it back over 20 years ago. And he has served as the FDMAS Alliance's Medical Advisory Council chair for over a decade, and he's currently our acting scientific advisory chair. He is the go-to expert for everyone in the community, patients and doctors alike, and it's my honor to be introducing him here today. Thank you.
Thank you, Cindy. My mic is on. You guys can hear me? Great. So thank you, Cindy, for that introduction. I really appreciate it. Thank you for the amazing work you've done with the $1 Million Bike Ride. Raising $1 million is a big deal, and I'm really, really impressed. And thank you for doing that so much. The work that you're doing, that this funding is doing, is having a major impact on the field that we're going to hear about today, including the simple fact that it's drawn these amazing investigators into the area of research on FDMAS. I also thank the other board members who are here and the members of the Scientific and Medical Advisory Committee that are here as well. And mostly I thank the parents of the patients and the patients who are here. I don't know if you recognize how much of an effect your dedication to your kids and your families have on us. It really is, to a large extent, which drives what we do, and it makes what we do that much more meaningful. So thank you as well. So I'll stop there and introduce Dr. Julia Charles. She's one of the newer investigators into the area of FDMAS, and it's because of this funding that she's here. And so this really speaks to the importance of this funding and this work that you guys do, because we need more people like her to push the envelope forward. Julia is at the Harvard Medical School. She was trained as a rheumatologist, and she's an MD PhD. She's one of these rare breeds of clinician and scientist that are so important to this field. Her focus is in bone diseases, osteoporosis, and rare diseases, as you'll hear about. She sees patients with Paget's and fibrous dysplasia, and she's going to talk to us today about her work that she's done with the funding that she received from the $1 Million Bike Ride. So with that, I'll turn it over to Julia.
Great, thank you so much. And thank you, Tova and Dr. Collins, for the invitation to speak. I'm hoping that you can see my slides now.
Now we see them, but they're small.
Okay. Any better?
Better, but.
All right, hang on one second. Let me see if I can get that better. Better?
Perfect.
All right, so I'm really delighted to have a chance to come and talk to all of you, because you're really the people who were responsible for the funding that allowed me to start this project. And it's a project I've gotten really increasingly excited about, and I'm continuing to look for funding from other sources and to be able to continue it. So I really come at fibrous dysplasia from a different perspective. I'm an osteoclast biologist. I have studied the origins of osteoclasts and how I'm very interested in how osteoclasts interact with other bone cells like osteoblasts and osteocytes. And that interest has really led me to be fascinated with diseases in which osteoclasts are part of a multicellular tissue. So diseases like Paget's disease, which is a classic rheumatologic disease where there's osteoclasts and osteoblasts all mixed up together. And fibrous dysplasia is one of these diseases. And so that's how I got interested in it. And the genesis of this project really came from seeing a talk by Dr. Yang and having read one of her papers and thinking, wow, here's this really interesting question that I want to ask about osteoclasts and what they do in fibrous dysplasia. And you can see here, I hope you see my pointer, but here's a picture, a histology picture of a fibrous dysplasia lesion. And you see these big giant cells with lots of nuclei inside of them. And those are osteoclasts stuck in the middle of the lesion. And what are they doing there? And what are they at all important for the fibrous dysplasia pathology is what kind of the question that drew me into this field. So I think some of you may know that fibrous dysplasia, the fibrosis part of the fibrous dysplasia is bone lesions, is this mix of early bone forming cells called osteoblasts, and that the tissue itself is mosaic, meaning that there are cells with the GNAS mutation that causes fibrous dysplasia, as well as cells that are genetically normal but are acting abnormally in that they're proliferating and being part of this collection of tissue in a place that should have bone instead of tissue, instead of a soft tissue. And the fact that the lesions are mosaic is very different from from what you think of as a classic cancerous tumor where all the cells are mutant. So this mosaic is a very, mosaicism of wild type and mutant is an interesting aspect of the disease. And then mixed into this mosaic lesion is my very favorite cell, the osteoclast. And I think it's clear that mosaicism, that the mutant cells are not the only part of biology that's important for generating these lesions. And work from Pam Roby's group at the NIH first showed that the fibrous dysplasia bone lesions are actually, she was the one to really show that the lesions themselves are mosaic and this mix of mutant and wild type cells or normal cells. And she actually showed that the interaction between the wild type cells and the mutant cells is important for generating the lesion. So if she took a lesion from a human and isolated the cells and then put them into a mouse to see if the mouse would get a fibrous dysplasia lesion, only when she took mutant plus wild type cells together did she get a lesion. So mutant cells on them alone were not enough. So this tells us that it's not just the cells carrying the mutation that are important for forming these lesions. It's something about how these cells interact with other cells nearby, cells that look similar but are genetically normal. And how also potentially how they interact with osteoclasts, and the interaction of these cells is needed to create the bone lesion. So my kind of big picture thinking is if you could understand how these different types of cells communicate to each other, you might understand what are potential targets for treatment for fibrous dysplasia bone lesions. So why osteoclasts? I mean, you might just think, well, osteoclasts are just, bystanders in the lesions, but what the kind of motivation for looking more carefully at the role of osteoclasts comes from studies in humans, actually. And the first very dramatic case report of using a drug called denosumab to block osteoclasts and looking at the effect on fibrous dysplasia. So denosumab, just to review, is a antibody that's administered to humans, and it blocks a key growth factor called rank ligand. And rank ligand is essential for taking precursor cells and turning them into osteoclasts. So if you get rid of this rank ligand, you don't make osteoclasts. And Alison Boyce, along with Dr. Collins, back in 2012, reported this really dramatic case where a child with fibrous dysplasia, bone lesions that was rapidly growing, was treated with this denosumab and had improvement in the rate of growth of the lesion and relief of pain. And one thing they showed, and has been shown by other investigators as well, is that the lesions themselves have very high expression of this rank ligand growth factor, which is probably why there are so many osteoclasts there, because they're promoted by all the rank ligand in the environment of the lesion. And since this case report in 2012, there have been 9 publications, either case reports or series of patients with fibrous dysplasia, where there's been reported benefits in terms of pain, slowing of tumor growth, and improved bone density at the lesion area. So clearly blocking rank ligand is doing something in this for the bone lesions. And people have tried to model this in a mouse model, and there's actually 2 reports now showing that blocking rank ligand, as shown here in a mouse with fibrous dysplasia, where every cell in the mouse carries the mutation, that blocking rank ligand was effective compared to if you look at a control treated mouse. So there's evidence both from mice and humans that blocking rank ligand and getting rid of osteoclasts is effective at decreasing the lesions in the bone. But my question was, why? Why does that do? And what are the osteoclasts doing? So we really thought about it in the schematic, which is actually from the grant that I wrote that allowed me to get the Fibrous Dysplasia Foundation funding to actually start this project. So we hypothesized that the cells in the lesion, these early osteoblastic cells, are making lots of rank ligand. That's recruiting the osteoclasts, but the osteoclasts in turn are talking back to the osteoblastic cells. And that if you either got rid of rank ligand on these cells, or if you blocked osteoclasts, you might disrupt this signaling. And then we want to find out, and clearly blocking this rank ligand has effect on lesions, but why is that happening? What is the effect of doing that on wild type cells, on mutant cells in the lesion? And can we understand how this is changing the behavior of the cells? And then we also set out to ask, is it important that these cells are expressing high levels of rank ligand? Is that important for the pathology to develop? And in order to answer these questions about specifically how the blocking osteoclast and rank ligand affects mutant versus wild type cells, we needed a mouse model in which you could easily tell apart, in which the lesions were mosaic and not just all carrying the mutation, but also where you could tell apart the wild type and the mutant cells. And this is just sort of summarizing the two questions we were asking. So we turned to a model that Dr. Yang's group very kindly provided for us. And this is a genetically modified mouse where at birth, the mutation, the GNAS mutation is inactive. And there's also a tracer, a lineage tracer that tells you, allows you to look at specific cell lineages. That's called tomato, which is a red fluorescent color. And at birth, neither of these are active. But if you treat the mouse with tamoxifen, a drug, you can then activate the mutation and you can activate the red tomato tracer in just very specific bone cell precursors. And you can compare those mice to mice in which the GNAS is wild type, but the tomato tracer is there but inactive. And in that case, after tamoxifen, these mice don't have the GNAS mutation, but they do have the red cell. So you can compare what happens to the same cells, whether if they carry the mutation or if not. And you can see that in our lab, we were able to reproduce the fibrous dysplasia lesions that Dr. Yang's lab had initially reported back in 2017. And you can see this area of kind of fibrotic material right along the cortex. And this is an early lesion where we're actually looking at these mice at a very young age, at 21 days of age. And that's compared to a wild type mouse here, where you see lots of hematopoietic cells, the blood cells, but not the fibrosis. So, if you look, and this is a figure from Dr. Yang's paper, which is actually the figure that got this project going in my mind. So here where you're comparing the mice where the red cells are mutant and the mice where the red cells are wild type, shown graphed over here, and you count up all the cells that have this unusual early osteoblastic marker called SMA, and you say, are those SMA cells wild type cells or are they mutant cells? So you can see that if are cells that are coming from where the tomato was induced, and you can see in the wild type, in mice that don't have the GNAS mutation, most of these early osteoblastic cells are coming from the lineage that had the tomato turned on. But in the presence of the mutation, there's a huge increase in these wild type cells, or the cells that didn't come from the induced, where they don't carry the mutation. And that suggested that there was non-cell autonomous communication. That means that the wild type cells are somehow being influenced by the presence of the mutant cells to expand this early osteoblastic phenotype that is not characteristic of osteoblasts. They very transiently go through this early stage where they express SMA, but you've got this huge accumulation of cells that are wild type but are not supposed to be at this early osteoblastic stage and are contributing to the fibrosis. So in our first question, we said, does blocking rank ligand decrease the bone lesions in this mosaic model? Because remember, it had been shown before, but it had been shown in mice in which every cell in the mouse's body carried the mutation. So we didn't know whether it would work in the model in which the cells are mosaic. So we took mice with the GNAS, where the GNAS mutation is inactive at birth. We gave them tamoxifen, and to induce the expression of the mutation in just some cells, not all cells of the body. And those cells that carry the mutation also turn on the red tracer. And at 21, and at the same time, we gave them rank ligand or control. And we said, is there a difference in the fibrotic lesions? These are the things very basic questions we wanted to ask with this pilot funding. Is there a difference in fibrotic lesions? And is it the mutant cells that are affected or are wild type cells affected? So We were very pleased that blocking rank ligand does increase the fibrous dysplasia. So this is a control treated mouse versus a mouse treated with the anti-rank ligand. And you can see that in the anti-rank ligand, the fibrosis tissue is not any different from wild type mice. And this just brings up, this graph here brings up a couple of, maybe there are problems with the way we've did our preliminary data, or maybe there are benefits. So there's one issue is that there's a lot of variation between individual mice. You can see here in the fibrosis, and we really find that there's a lot of variation among animals in terms of how many, what degree of mutation, accumulation of mutant cells we get and how much fibrosis we get. But maybe that's a benefit because that's more like real life and how there's a big phenotypic variation between different patients. The other issue is that we looked in very young mice, so it's very hard to tell what's normal early osteoblast growth versus fibrosis. And so we're trying to repeat all of these experiments that I'm telling you about in older mice at this point. But we were very happy that we basically got the rank ligand treated mice looked very much like the wild type after treatment. And so then we wanted to say, okay, what happens to wild type cells versus mutant cells? Is it the wild type cells that are being affected by the treatment? Is it the mutant cells, or is it both? So if we use the tomato tracer and then antibodies to specific markers that are known to be at various points in the osteoblast lineage, we can show that the red cells here are that marks the cells that are mutant. And if we use SMA, this early immature osteoblast marker, we can say if they're purple, they're early osteoblasts. If they're purple and red, they're early osteoblasts that are mutant. And if they're purple, just purple, then they're wild type. And then blue it shows the nuclei that helps us make sure we're actually counting cells and not debris. So if you block rank ligand, you can see here's a control mouse and one that's treated with anti-rank ligand. You can see that there is no difference in the number of mutant cells per tissue area. And then you can see the rank ligand treated mice have lots of extra bone. So we thought, well, maybe we're analyzing this the wrong way. So we went back and counted them per bone surface, and that also was not different. So we don't think that it's affecting the number of mutant cells or causing them to die off or anything like that. But conversely, this tells us that since there's less fibrosis, the rank ligand must be inhibiting the recruitment of the wild type cells to the lesions. And we're currently trying to figure out the best way to actually differentiate between wild type cells and to specifically mark the wild type cells so that we can count them and not just infer that they must be decreased by the treatment. So what about the actual characteristics of these cells? So we also looked at whether or not what happens to, in the mutant cells, what happens to, they may not go away, but do they change their phenotype? Do they change how they behave? And so we looked at what percentage of them express SMA, which is this early osteoblastic marker that characterizes immature osteoblasts. And we can see that that is decreased by the rank ligand treatment. And so we said, okay, well, maybe they're becoming more mature. So we looked at Osterix, which is a slightly later marker, and that was not changed. So we're actually wondering whether if we look at very mature markers like osteocalcin, will we see an increase in the mutant cells that have become mature? And we kind of hypothesize that we may, because in fact, we see so much extra bone in these mice, suggesting that many cells in the lesion have become active bone-forming cells. So this is sort of a TBD, but we're actively working on that. And then the next question was, is it important that the rank ligand is made by the mutant cells? Is that an important part of the pathology? And so now we make these mice that are even more complicated and have yet another genetic mutation. So we either have mice that have the GNAS mutation and tomato in an inactive form and have wild type gene for rank ligand, or we have the same mice in which we've conditionally inactivated the rank ligand. So here at birth, they have normal rank ligand, so their skeleton develops normally. We give them tamoxifen. And in this case, the cells that have the GNAS mutation activated and have the tomato tracer no longer have rank ligand. And I was so hoping we would get pathology from these mice before this meeting, but unfortunately the sections are still being cut. But just by micro CT, we think there may actually be a difference. So we were now looking at 6 weeks of age to try and get away from some of the problems we had with the 21-day-old mice. But you can see in the wild type, where rank ligand is wild type, there's all this kind of weird, fuzzy, sort of ground glass appearing bone characteristic of a fibrous dysplasia lesion along the cortices. And it's much less prominent in the mice that lack rank ligand. Now, this took us much longer to do than we expected. And so we're still beginning to, we only have a few mice in each group and we're beginning to do the histology, but we're really pretty excited about at least the early micro CT results. So the takeaway points from the pilot project so far is that we can show that blocking rank ligand decreases fibrosis in this model, in which we can actually, we know it's mosaic, so we can really try to tease apart how all the different cell types are interacting with each other. And this further supports a role for osteoclasts in promoting the fibrous dysplasia bone lesions. And we're most excited about the idea that we can use this mosaic model to tease out the effect of blocking rank ligand on the mutant cells versus the wild type cells. And we're currently doing additional cell marker studies. But what we'd really like to do in the future is to grind up the bones, take out the cells that have been, whether they've been treated with rank ligand or not, and then do something unbiased, like sequencing the mutant cells before and after treatment, sequencing the wild type cells before and after treatment. And then I think the significance of the rank ligand expression by mutant cells, we're hopeful that this will actually be an interesting angle, but it's really TBD. Okay, so with that, I'd like to give a big thank you to the funding that started this project off, and to Dr. Yang, who was instrumental in providing the mouse model, and to Orphan Disease Center and $1 Million Bike Ride for all the great stuff you guys do, and to Renee, who was the postdoc who did a lot of this work. Thanks.
Thank you, Julia. That was very nice, and it was very clear. That was really complicated stuff that I think you did a great job of making understandable to people. And Tova, how much time do we have for questions right now?
I think we comfortably have 10 minutes. In between, maybe 7 would be safer, but.
Okay. So, well, if we're running long, just let me know. Anyway, Cindy has been posting some questions that came from people, I guess. And there's one really good question to start with here. And the question is this, Julia, and it is there anything demonstrated from your work? No, that's not the one I wanted to ask. Sorry. It's this one. Julia, do your studies reveal any distinction between treating children with active growing lesions versus adults whose lesions are more dormant?
So that's a really awesome question. And, so far we have only treated mice starting the treatment at an early age when the bones are still growing. And I think you could see by the one image, and actually maybe I'll just share the screen and go back to that. Treating young people or animals with anti-rank ligand is not a completely benign thing, right? Because you see how much bone has developed. And I don't know if you can appreciate, but the bone is actually substantially shorter. So it really inhibits the longitudinal growth at least in animals. And I'm assuming with persistent use, it would do the same in humans. And so we've really only treated young animals. We're starting right now treating animals who've already developed the lesions and finished skeletal growth. And that will maybe inform the answer to the person's question a little bit. But I think I don't know whether the mouse model, if you let them age long enough, whether they would ever get to that point where the lesions are more kind of quiet, and people have shown that the number of mutant cells actually decreases in some of the lesions that are not actively growing, and whether there are still osteoclasts in those lesions. I don't know the answer to that, and actually I'd be really interested to know whether maybe Dr. Collins knows in lesions that are not actively growing, that are more quiet, have fewer mutant cells, are there still osteoclasts in those lesions?
Yeah, so the number of osteoclasts is in lesions is proportional to the relative, I would say, activity of the lesion. So the more active the cells are in dividing, the more dividing cells there are, the more osteoclasts there are. So, and as lesions, and what we're all talking about here is that it appears to be the case in patients with fibrous dysplasia that there's a rapid period of accrual of bone disease early in childhood, and that really is plateaus later on in life. And so in this plateaued period, where we kind of see it as, the damage is done to some extent, there's less cell growth, there's less cell division, and there's less osteoclasts.
Which would argue that maybe the lesions would not be as responsive.
It argues they wouldn't be as responsive, but the one thing I could say is that, in some older patients who have fibrous dysplasia and in whom pain is a major issue, those patients do seem to benefit a lot with getting pain relief from treatment with denosumab. These haven't been, controlled studies, and they haven't been well done, and, it remains to be seen, but it does appear to be the case they have less pain. I know in that first case that you showed of that boy who was taking narcotics on a daily basis, he was a child and not an older patient, but still, he went off pain medication completely with the treatment of denosumab. So somehow, we're not quite sure how fibrous dysplasia causes pain, but it does appear to be the case at this point in time that blocking rank ligand with denosumab decreases the pain.
And I'll just say there is a growing literature in the osteoarthritis field about osteoclasts potentially being mediators of nerve growth into areas of painful bone. So it's possible that even that's the mechanism for pain rather than any actual change in the lesion size. So I think there's a lot still to be learned.
There's a lot to be learned, and these mouse models that you're working with, that Yingzhi, Ed Xiao, and others have developed are just so useful for trying to figure this stuff out. I mean, as you can see, with the slide on the page here, these mice volunteered their leg to be studied, and we can't do that with patients, but we can learn a lot from these models, and this is the sort of thing that this research is so important for, and this funding is so important for. There was, let's see, that was one question. This is a question from Cindy Brandt. Is there anything demonstrated by the fact that the tamoxifen brings about the mutant cells in the mice?
So just make sure I understand. Do you mean that are we sure that the tamoxifen really does induce the mutation? Or
I think what she means, and I think this is really kind of a technical question. These mice have been engineered so that they have a receptor for tamoxifen. So obviously mice don't normally see tamoxifen, but when this drug is introduced, it's like an on-off switch that turns it on. So it's a technical aspect of what's intrinsic to these.
Right.
I think a lot of people are familiar with tamoxifen because it's used to treat breast cancer, blah, 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 but that's all separate from what Julia has done here. This is and Yingzhi did that. This is a technical on-off switch that's been introduced into these mice specifically for this purpose. Does that make sense, I hope?
Yes, thank you. I just wasn't sure if the tamoxifen itself had any significance with regard to
it's really, I mean, I could have put out the whole genetic thing about how the tamoxifen receptor is only expressed in early osteoblastic cells and only briefly, and then you add tamoxifen very briefly, you turn the mutation on in just a few cells of the early osteoblast lineage. But but it's a great question, and I'll try and figure out how to make that clearer in future talks.
Yeah, I mean, these are really complicated models and complicated stuff, and it's hard for scientists and doctors to understand sometimes. So I think you did a great job, Julie, of making this clear for people.
Thanks.
Now we have a question from one of our ringers in the audience, Ed Shao. Dr. Shao, hi Julia. I seem to recall a prior slide suggested a decrease in FD with the anti-rank ligand treatment. The CT suggested that the bone doesn't change, but might be higher, but that the fibrosis is decreased.
So
Nice
I
talk,
think,
by the way.
I think, thanks, Ed. I think what Ed's talking about is that the bone, maybe he means that the bone appearance doesn't change, but there is lots of extra bone. And you can see that both on histology and on the CT, you can see this kind of big white area. That's all extra bone, or bone that isn't normally there. And when you look at that bone, the bone, at least on light microscopy, looks relatively normal. And it's just very, very thick. Every little piece of bone that's normally there is much thicker than it usually would be.
Here's a question from one of our parents, and tell me, Tova, when we're out of time. This is from Amon. My daughter has FD in the skull base / temporal bone and is losing hearing in the left ear. The tumor put pressure on the 7th and 8th nerves. She's still normal, but what is the progress? What will happen if the pressure continues on those nerves?
So, that is a really wonderful question. And I think I would have to defer to one of the clinicians who's more experienced in treating fibrous dysplasia of the head and neck than I am, which I think would be Dr. Collins. I don't.
Yeah, so this is a very specific question, and it's really hard to give a specific answer in this sort of format. But I'll do my best, and what I can say is that generally, while fibrous dysplasia is very common in that part of the skull, and very common to encase the 7th and 8th nerve, it's very uncommon for patients to lose a significant amount of hearing. It's not uncommon to lose a little bit of hearing, but to lose the amount of hearing, for example, that you either go deaf or require a hearing aid, that's very, very uncommon. So generally speaking, it's unlikely that she'll have significant hearing loss as a result of this. But again, it's difficult to respond to your specific question. And maybe afterwards you can email one of us and we can get more specifics and maybe try and connect with your doctors or connect with other doctors or something like that, which is another wonderful service that the FDMAS Alliance offers. They have a very nice registry of highly qualified doctors that you can talk to.
I think, Mike, at this point we should transition to Dr. Ying's presentation. There will be time at the end for as long as people are able to stick around for more questions, but I want to make sure we give space for that.
Thank you for your attention.
Great job, Julia. Thank you. So we're going to transition now to Dr. Yingzhi Yang. Dr. Yang is the Dean of Research at the Harvard Dental School. She is a professor of developmental biology. She is a major leader in the field of bone research. We are so lucky to have her here today. We are so lucky to have her involved and dedicated to this research. The mice that she have made that model this disease, which Julia just used and have talked about, have had a major impact on the basic science in this disorder. So So glad you're here today, Yingzhi, and thanks for the work you do.
Dr. Yang, you're still muted.
Can you see my slide?
Yes, but now they're in presenter mode. We can see the slides and we can hear you.
Okay, great. Thank you, Michael, for the generous introduction. It is really a great pleasure to be a member in this research field, and hope our work will contribute to the eventual cure of this devastating disease. So I have to give a lot of credit, or major credit, to a previous very talented postdoc fellow, Jean Rigard, who is now in charge of clinical trials at the Novartis Research Institute in Boston. And it's really nice still being able to talk to him from time to time. So at that time, Jean was interested to ask what is the molecular mechanism underlying this disease, McCune-Albright syndrome, which is due to mosaic mutations in GNAS. Julia already told us what that means. And one of the clinical manifestation is fibrous dysplasia, which is a bone phenotype in the patient. There was bone overgrowth and fibrosis. Here is a biopsy, and I believe was done by Michael. The novel healthy people have this nicely mineralized bone and with marrow in it. But the patient exhibit very little bone marrow, hematopoietic cells, and then ossification shown by this black von Casa staining was much reduced. So many years ago, Jean decided to look into the signaling pathway of Wnt beta-catenin, which plays a major role in osteoblast differentiation. What he found is that the mutant cells had this highly activated Wnt beta-catenin signaling, and that is at least partially responsible for this poorly mature osteoblast and poor mineralization of the bone in the patient. What he found with the help of Michael and Pam Roby at NIDCR at that time is that in this in vitro osteogenic differentiation assay, the wild type cells can form nice bone modules shown by von Casa staining with this characteristic marble-like pattern. The mutant cells couldn't do so, but if beta-catenin levels were brought down by knocking down experiment, there was this progressive rescue of bone formation, and therefore this characteristic marble-like staining pattern. So from that, the conclusion at that time was that this mutation in G-alpha-S, which locked this protein in an activated state, led to upregulated and sustained beta-catenin activity, and therefore blocked osteoblast differentiation and therefore mineralization. So in order to further understand the molecular mechanism and to test potential therapeutic strategies, we decided to generate a mouse model that will allow us to accurately mimic FD condition in human. So Julia had already talked about what this mouse model can do. And basically we put one of the human mutations identified in human FD patients into the original GNAS locus in mice in a way that this mutant expression can be induced in the tissue and the cell specific manner. As we know, this disease is a mosaic, meaning that it cannot be inherited in germ cells. And why is that? After we got this conditional mutant mice, we found that if the mutation is inherited in the germ cells, it is going to cause in mice, we call it embryonic lethality. In human, that is lethality in fetus. So this mutant could not be inherited in germ cells, but the genetic strategy allow us to express it in skeletal progenitor stem cells. And we found if we do so, we can have mice that show phenotypes very similar to FD patients. As you can see here, after micro CT scanning, there was this enormous bone overgrowth, and although the mineralization was reduced, shown by this reduced von Casa staining. So what I want to say here, just like what Michael had alluded, mouse models are very useful and they are essential for disease studies for at least the 3 reasons. First, it is essential for mechanistic discoveries, at least at the molecular, cellular, and the physiological levels. And 2, it is essential for the identification of druggable targets. And third is it is essential for drug screening and therapeutic tests. So with this in mind, we decided to look more into the cellular and the molecular mechanism. And Julia had already talked about this so-called non-autonomous effect, or how the mutant cells could influence the surrounding genetically normal cells and to show abnormal defect. And I'm not going to repeat that. And basically, we found that the mutant cells can have profound influence on their surrounding wild type cells, and that influence may contribute to the disease prognosis or disease phenotypes. What I want to say is that from the previous study with the human FD cells, we find Wnt beta-catenin signaling is important. So now with the mutant mouse in hand, we decide to further test whether this is still true in this whole animal model. And the answer is yes. So here's the FD mutant model with bone overgrowth and increased fibrosis. If we reduce Wnt beta-catenin signaling by reducing or removing part of its receptor called LRP6, and now that phenotype is improved. And we also can use a drug that can reduce Wnt signaling levels. And in this in vitro osteogenic assay, we find that this drug, again, can improve osteoblast differentiation shown by von Casa staining. So with the help of this foundation, we tested the effect of this drug, it is called LGK974, in vivo in the mouse model that we generated. And as you can see here, this mutant mice was a little smaller. LGK974, if we reduce the Wnt signaling, that will make the wild type control actually a little bit smaller because it could reduce bone growth. However, if we just compare the mutant cells treated and non-treated, there isn't much bone defect, and more importantly, and I think we have to repeat this with more numbers, is that there was this significant bone shortening in the mutant, but after this drug treatment, the bone shortening is rescued to a large extent. The cartilage growth plate looks better, and then bone growth also looks much better. I should also mention that FD patients show profound skull defect. Skull bone formation was quite irregular. In some places, there's more bone formation. In some other places, the bone formation was very little. There was bone cyst and then fibrosis. And we find with the mouse allele that we have generated that indeed the similar phenotypes can be seen in the mouse mutant. And again, that was due to upregulated, at least in part, due to upregulated Wnt signaling. And we use this in vivo LAC-Z staining here. If there's more Wnt beta-catenin signaling, the mouse will have more blue staining. So in the mutant, as you can see here, there's a lot more blue staining compared to the wild type control. So in skull bone formation, the FD mutation also leads to more to higher and sustained Wnt beta-catenin signaling. And then the next question is that if we bring down Wnt beta-catenin signaling, can we make this phenotype better? And the answer is yes. And we treated mice with the same drug. As you can see here, there was much less bone formation or mineralization in the FD mutant skull compared to the wild type control. But after this drug treatment, bone skull formation in the mutant was much better. So Wnt beta-catenin signaling is maybe still a viable target for treating this patient. However, we know that Wnt beta-catenin signaling is essential for normal bone formation. And there's literature showing that with the treatment of this drug, normal bone homeostasis is affected. So we are also in the middle of looking for additional targets that may potentially benefit the patient. So one of the targets that we identify is YAP. So in contrast to beta-catenin activation, we find that in the FD mouse model, there was a reduction of YAP signaling or YAP protein activity. And this is shown by this immunohistochemistry, the protein level was reduced. So what is YAP? And like Wnt beta-catenin signaling, YAP is in the signaling called Hippo. And this signaling pathway is also very much important in cell proliferation and cell differentiation. It has been shown to be very important in bone formation. So therefore, we think and this pathway may worth further investigation, and I hope we'll have more data to share next time. And with this, I would like to thank our collaborators, of course, my co-colleagues, Pam Roby at NIDCR. Thank you for the collaboration, and the collaboration is still ongoing. And also other colleagues at the NIH, Lee Weinstein, our long-term collaborator in other area, which is opposite to this disease, the loss of function of GNAS, Fred Kaplan and Elaine Shaw at UPenn. The work is carried out by previous student Roshi Shi and previous postdoc Sanjoy Kam, and now it's continued by current postdoc fellow Chen Chong and technician Andrea Orozco. And I should thank the funding agents, the UPenn million dollar bike fundraising effort, and also NIDCR and NIAMS for grant.
Thank you, Yingzhi. That was really good. As I said, Yingzhi has been a major force in this field in creating models to allow us to study this disease and others. And as you can see, they're incredibly complex but incredibly informative. While we're waiting for questions to come in, and we only have about just a couple minutes left for questions. One of the things I wondered about, and I don't know the answer to this, do you know where the field stands in terms of drugs that would be available to target the Wnt pathway or the YAP pathway? I know that early on, these were fairly toxic drugs that were only used in cancer, but they'll probably get better. Where does that stand now and as a potential treatment for FD?
Yeah, so as far as I know, the drug we use, LGK974, is probably the best one in terms of Wnt beta-catenin inhibition. And yes, you're right. It was initially developed as a cancer treatment drug. But we all know that Wnt is essential for bone homeostasis, and they tested in mice and find that it will cause marrow, what to say, over differentiation of adipocytes in the marrow. So there and reduce the bone mineral density. So that is not good news. But on top of that, I don't think I'm aware of other drug that can be used in vivo. This one so far is probably the best.
No, that sounds good. And I think what's hard, I mean, what all the parents always want to know is, where's the drug that I can give to my child? And this is where that drug starts with. You start with a discovery like you made of the importance of this pathway, and then you find a drug that targets that pathway. This drug is super toxic. The drugs get better, the ways to administer them get better. It's a long road, but the road starts here, and it's through work like you do, and then that translates into seeing patients like the work that Dr. Boyce is doing here. So, I know it's always slower than patients and families want it to be, but the amazing thing is that for this rare disease, a lot of inroads have been made in understanding it and developing therapies. So there's hope.
Yeah. So, first we need to have the science there, and then we can hope there's continuous improvement from both basic science laboratory and also the industry. And the same thing is for YAP. So I don't know whether it is better to target the YAP. So far, we think maybe we need to increase YAP activity. And then there is no good drug for YAP enhancement. And again, YAP enhancement may have pretty bad effect if you don't do it right.
So we've actually run out of time, but I did on the last minute, Julia, Dr. Charles, you put something in the chat about Wnt inhibitors. Do you want to give us a little blurb on that?
Oh, I know that there a company called SamuMed had developed one specifically for osteoarthritis treatment, and it was in clinical trials. I don't know, is that the same one that you're using? But you're using one that was developed for cancer.
Osteoarthritis.
Yeah, so there, I know there is that there are. I'll have to go look that up. But 2 or 3 years ago, they were recruiting patients for OA trials to use a Wnt inhibitor specifically to look at progression of OA. Maybe that would be a less toxic inhibitor.
Yeah, I'll look into that. Yeah. Yeah, so Wnt signaling in OA is also complicated. As you may know, too much and too little isn't good.
Tova, we're out of time. What should we do?
As Dr. Yang, Dr. Charles, are you coming up against something that you'll need to go to? No, so I think we can stay and answer questions. I do want to make note of something just in case we are going to lose anybody here. That we just recently in the last 24 hours were able to finalize an important meeting for FDMAS research, and we wanted to share that here. And that's that on April 5th at 4 p.m. Eastern time, we have guest speaker Dr. Cecilia Duboucher, who's a program manager with the peer-reviewed medical research program. And she's going to come and review the PRMRP funding opportunities with the congressionally directed medical research program. We'd highly encourage researchers who study FDMAS to attend and gain insight into the PRMRP process in preparation for 2022 funding opportunities. Again, we've just finalized this. We really hope people are able to attend. Reach out to Adrian to RSVP and please share widely. So that, again, that's for all researchers. But as long as folks are able to stick around, I think we should continue to take questions.
Yeah, I just want to second your support of that. I know that this is an unusual funding mechanism. Not so many of us are so familiar with it, but it is a lot of money, and there's some real peculiarities about the process. And I know some of you have been involved in the past, and we're trying to gain better insight in how to tap into this significant fund of money for FDMAS research. So I think this would be a good thing for all the investigators to attend if they can. Tova, I actually do have to run, which I'm sorry to say. So maybe you can take over the moderating for me and I'll run. And again, thank you all. Thank you, Julia. Thank you, Yingzhi.
Thank you.
Thank you so much, Dr. Collins.
You're welcome. And I'll see you guys later. Bye-bye.
Bye. So I will do my best to read these questions, and please excuse my pronunciation. Let's see here. So we have a question, and actually I'm going to invite Dr. Chow. Do you want to come on and ask your question, and others, if you care to come on video? You put it in the chat. Are you still with us?
Yeah, and I tried to answer Dr. Shao's question from earlier just in the chat, but I think he had one more for Dr. Yang.
I can read it if he's, let me find it here. This is great talk. Have you seen toxicity from the LGK treatment of the mice, either in FD or in the wild type mice? I think the human clinical trials also showed problems with tolerance.
Yeah, that's a great question. So we did see smaller bones after treatment, but since the bone was abnormal in the mutant, and after treatment, actually the bone looked not as bad. So I think that was encouraging. But I agree because this is a mosaic disease. The mice wasn't so mosaic, so we can't use something that is going to make most of the normal bones in the patient worse, and then just a small part look better, a little better.
And then we had some questions come in with our registration, and many people, as Dr. Collins mentioned, really want to know how far are we away from seeing this in, at our doctor's office. Where are we on that path to a treatment? I know it's a really challenging question. Do either of you have insight into that process?
Well, I guess I would just say that, denosumab and osteoclast inhibition is already in use. And I think hopefully there will be trials eventually to show in a more kind of clear way the benefits, but we don't know exactly how best to use it, what doses, what interval, how long can you use it safely in a growing child. And Ed may have more information about whether there are trials planned. There have been trials in giant cell tumor, which is another similar kind of mixed kind of cellular tumor or growth in bone. And denosumab was very, very beneficial there, but we don't have that same kind of data in a systematic way in fibrous dysplasia. Though maybe someone's trying to get a trial together. Ed might know about that.
You sort of alluded to this, Dr. Charles, and I think also put something in the chat. There are different funding mechanisms beyond the $1 million bike ride, and it can be a real challenge to find the right place for your research. Can you speak to the importance? Because relatively speaking, the $1 million bike ride grants are smaller than your NIH or your Department of Defense funding that we spoke about earlier. how important is the $1 Million Bike Ride and Team FDMAS
Right.
fundraising for your work?
I mean, so for a junior person like myself who had never been in the field before, right? I would never have gotten funding from NIH or DoD. And even with the preliminary data generated by here, I actually got very positive feedback from both NIH and DoD but didn't get funded, although of course I'm going to keep trying. But it allowed someone to come into the field that had not been there before. And I think you need that to keep kind of rejuvenating the thought process. You need people from outside to help kind of bring new ways of thinking about the problems.
Yeah, I would echo Julia's comment. And I also felt this $1 Million Bike Ride Foundation or fundraising effort is essential to bring the community together. Now Julia and I are working together, and hopefully someday we will be able to maybe form a team to get major, much larger fund to do more work together, such as this PRMRP from DoD.
Looks like we do have a question from that parent again. Eamon, do you want to ask your question?
Yes, Tabu, thank you very much. My question is regarding my daughter had a fiber dysplasia in temporal bone. So the tumor are putting too much pressure on the 7 and 8 nerve. So I asked the question in regards that the progress about the growth of this tumor, if it comes too much big, so what is the situation in this 7 and 8 nerve? And she's still normal till now.
Did either of your research look into this 7th and 8th nerve and this type of question?
No, and I can jump in as a clinician. I'm going to put on my clinical, not my science hat for a second. And Ed, if Ed is listening, I'm not sure he's actually still on the call, but if he is, he can jump in too. But definitely any situation in which there's chronic pressure on a nerve, if the pressure starts to cause the nerve to be inflamed or not get enough blood, it can cause damage to the nerve. So I'm hopefully, and I'm sure your physicians are watching that carefully. But it's very difficult to speak to your daughter's situation exactly because I'm not her physician and I don't know exactly her situation. But you hopefully you have a good team, someone who's working on the fibrous dysplasia part, a neurologist or ENT, oral maxillofacial person who, for a while there was one of the OMFS people from MGH was on the line. Like you need people who really are familiar with that area of the body and the very complicated anatomy there to know at what point do you need to do something to relieve the pressure on the nerve. And whether that's surgical or something like denosumab, I would defer to your daughter's physicians.
Dr. Charles, you had asked a question in the chat about the WNT inhibitor. Did we have a chance to touch on that? I think that's
right. Yeah, we did. We talked about that already. Yeah.
So another question that came in was general in terms of your research. What would you say are one or two of the biggest challenges that you face in getting to that next breakthrough in your FDMAS research? Maybe each of you could answer. Dr. Yang, do you want to lead us off?
Yeah, so I, of course we need more resource, right? So, a lot of experiments, and I think it's going to be more informative, but they are more expensive. Julia brought up the necessity of RNA-seq, for example, and not just the bulk RNA-seq and single cell RNA-seq and all this. We need to have much better understanding of the cells involved in this disease. So I guess, without more resource, it's difficult to get better or deeper understanding of this disease. And also, and once we have candidate target, we also need resource to test out or screen drug, right? Develop screening assays. So, and additional mouse models. I guess a bigger, I think we need to find more opportunities for major support.
I think for me, yes, resources is absolutely one issue, but there's also just the complexity of the disease itself, right? It's not a linear straight, if A is wrong, if A is mutated, then B happens, then C happens. It's a much more complex interplay between cells. And so I think it's just a very challenging scientific problem. And the models we have are great, but they're not perfect. I mean, I think I personally am biased towards using the mosaic model that Dr. Yang developed, because I think that's an interesting part of the disease that we really need to be able to understand, this kind of wild type and mutant cells talking to each other. But no model's perfect, and mice are not humans. And, I think that's kind of as a clinician, that's ultimately where I always get kind of hung up on, like, I can do this research and it's exciting and fun and I love it, but how keeping the faith that it's eventually going to help patients is sometimes really challenging. And honestly, the kind of support of patient groups is really important for keeping that faith, right? Because if you guys have faith in us, that helps us have faith in ourselves too.
This is, hearing you speak about your work is so important and so inspiring for us. It really, I hope you're seeing the messages of gratitude coming in. It really does mean the world to us in this community. One more question from our team captain, Cindy Brent Levin. As a community, is there anything we can do to help foster more funding for research outside of, the $1 million bike ride we're very committed to, and the Department of Defense funding? We really hope to see success for an FDMAS researcher in those areas. Is there anything beyond that we as a community could do to support this and bring more resources?
I guess I always wonder about industry and those companies that really specialize in orphan diseases. And, I can't remember all of the names, but there's like Ultragenics, which makes a drug for a very, very rare disease, bone disease, XLH. Like, would they be interested in helping out? So some of those pharmaceutical companies might be a good place to look. And I just don't, I'm not connected enough to that world to know how you would even begin to do that, but I'm sure there are people who do know.
Yeah, I would echo that point. I think we should engage pharmaceutical companies. And the other part, for me is that, I'm not a medical doctor. So getting access to human patient samples or cells would be important for our research as well. As Julia said, mice are not human patients. No matter how similar they are not. So I think having access to human patient samples and cells will be essential for us to do better research in this area.
That's actually really interesting. Like, so I'm also peripherally involved in a rare disease that has bone manifestations called tuberous sclerosis, and that community has actually generated a very large tissue bank and serum bank where they are very active as a community in recruiting patients' families to collect samples and store them locally. And then investigators can apply to use those samples for and that application, because the samples are precious, goes through a scientific advisory board, for example. You don't just get access if you ask, but it is a resource that is pretty remarkable that the community itself kind of helped spearhead and fund.
It's one of the things that I really love about the research update is I think every year it's generated new connections and new ideas, and certainly building relationships to pharma is, I know, something that we as an organization have been working towards. And if anybody here does know somebody who we should be in touch with, we would welcome those connections, and certainly exploring a biobank is something we've talked a lot about. Yes. Adrian, I see, is saying we'd love to have a conversation with PAG and with tumor fibrosis. Not seeing any other questions come in. I think we will close out for today. Thank you so much, Dr. Yang, Dr. Charles, for your work and for focusing your time and talent on this community. We are so grateful. For those on the call who are interested, Team FDMAS is gearing up again for this year and for the next round of Team FDMAS $1 Million Bike Ride fundraising. We'd love to have you join us. You can learn more. I can put this link in our chat for all of you. Thank you so much. And if you do have questions or comments, you can reach out to Adrian McBride. We'd be happy to hear from you. Thank you so much.
Thank you.
Thanks so much for
Thank you.
organizing.
Thank you very much.
Take care, everyone.